In this question, you have to identify the mechanism that causes a gene to move from one linkage group to another. Option A, translocation. Now, translocation is the process where a segment of chromosome moves to a different chromosome. So this can result in a gene shifting from one linkage group to another. Hence, it is the correct option. Option B, crossing over. It is incorrect because it involves the exchange of genetic material between homologous chromosomes during meiosis, which creates genetic recombination, but it does not involve a gene moving to a different linkage group. Option C, inversion. It is also incorrect because it refers to a chromosomal mutation where a segment of chromosome is reversed in orientation, but it does not cause a gene to move to another linkage group. Option D, duplication. It is also incorrect because it results in the repetition of a segment of a chromosome. So it does not lead to the movement of a gene between linkage groups. In this question, you have to identify the correct statement regarding a true breeding plant. Option A, near homozygous and produce offspring of its own kind. True breeding plants are those that produce offspring with the same traits as in the parent when self-pollinated. And these plants are homozygous for the traits of interest. That is, the genetic constitution is nearly uniform or so. Hence, it is the correct option. Option B, always homozygous recessive in its genetic constitution. This is incorrect because true breeding plants can be homozygous dominant or homozygous recessive depending on the trait. Option C, one that is able to breed on its own. This is also incorrect because this definition does not capture the essence of true breeding. Now, many plants can self-pollinate or even cross-pollinate, but being able to breed on its own kind does not ensure that they produce offspring identical to the parent. Option D, produced due to cross-pollination among unrelated plants. This is also incorrect because cross-pollination among unrelated plants introduces genetic variability, which is not applicable for true. In this question, it is given that a color-blind man marries a woman who is homozygous for normal color vision. So, what is the probability of their son being colorblind? Now, color blindness is a sex-linked recessive trait which is carried on the X chromosome. So, the man with color blindness has the genotypes XCY, while a homozygous woman with normal vision has the genotype XX. Now, from the Punnett square, it is evident that all daughters will be carriers but not colorblind, and all sons will inherit the Y chromosome from their father and normal X chromosome from their mother, making them normal. So the probability of their son being colorblind is 0%, as the mother does not carry the colorblind allele on her X chromosome. Hence, the correct answer is option C. In this question, it is given that genotype of a husband is IAIB and that of his wife is IAI. So, among the blood types of their children, you have to identify how many different genotypes and phenotypes are possible. Now, as the genotype of husband is IAIB, therefore the gametes that he will produce would be IA and IB. And for the wife, the gametes would be IA and I. So, after making the Punnett square, we can see that genotypes of the children are IAIA, IAIB, IAI and IBI. So, the phenotypes of the children, that is, children would be having blood group A for IAIA and IAI, blood group B, IBI, and blood group AB, IAIB. So total number of genotypes is equal to 4 and total number of phenotypes, that is group A, group B and group AB, equal to 3. Hence, the correct answer is option B. In this question, you have to identify a disease that is caused by an autosomal primary non-disjunction. Option A, Klinefelter syndrome. It is incorrect because Klinefelter syndrome is caused by a sex chromosome aneuploidy, that is XXY, and that results from non-disjunction of sex chromosomes and not autosomes. Option B, Turner syndrome. It is also incorrect because Turner syndrome is caused by a monosomy of X chromosome. That is, one X chromosome is absent, resulting in 45, the chromosome number. And this also results from non-disjunction of X chromosomes and not autosomes. Option C, sickle cell anemia. It is also incorrect because it is a genetic disorder that is caused by point mutation in the beta globin gene on chromosome number 11 and not due to non-disjunction. Option D, Down syndrome. Now, non-disjunction refers to the failure of the chromosome to separate properly during meiosis, and it leads to the gametes with an abnormal number of chromosomes. So Down syndrome is caused by trisomy 21. That means there is an extra copy of chromosome 21. And this condition arose from non-disjunction during meiosis. Hence, it is the correct option. In this question, you have to identify from among the following characters which one was not considered by Mendel in his experiments on P. Now, Gregor Johann Mendel did not consider the presence of glandular or non-glandular trichromes in his experiments on pea plant. Rather, his focus was on the 7 observable traits like seed color, yellow or green, then seed shape, round or wrinkled, then pod color, green or yellow, pod shape, inflated or constricted, flower color, purple or white, flower position, axial or terminal, or stem length, that is tall or dwarf. But trichomes was not among these traits. In this question, you have to identify the period during which Mendel performed his famous hybridization experiments. Now, Gregor Johann Mendel conducted his famous hybridization experiments on Pisum sativa between 1856 and 1863, and these experiments formed the basis of Mendelian inheritance, and it lays the foundation for modern genetics. And his findings were later published in 1866 in the paper entitled Experiments on Plant Hybridization. Thus, the correct answer is option D, that is 1856 to 1863.